DNA. You've probably heard of and seen it before. But what is DNA? What does it do? DNA has a double helix shape, resembling a twisted ladder. Each rung is made of nucleotides, of which there are 4 types: adenine, cytosine, guanine, and thymine. Each pairs specifically with its complementary nucleotide partner. Adenine pairs with thymine, and cytosine pairs with guanine. The specific arrangement of the nucleotides determines the unique genetic code of an individual, dictating traits and characteristics. DNA is found in the nucleus of a cell, specifically in the chromosome. Each human has 23 pairs of chromosomes, or 46 chromosomes, 23 from each parent. Chromosomes are made up of many DNA molecules, tightly coiled around proteins called histones, which support its structure. Without such tight packaging, DNA molecules would be too long to fit in cells, making them unable to accurately copy during cell division. The DNA is methodically organized into genes, which are the units of heredity passed on from parents to children. Simply put, DNA is the code of life. It contains the instructions necessary for an organism to develop, survive, and reproduce. It determines if your hair is black or brown, if a fruit fly has vestigial or wild-type wings, and if a flower is yellow or blue. These traits, also known as phenotypes, are expressed through a process called protein synthesis. Protein synthesis is the biological process by which cells build proteins using the instructions encoded in their DNA. The protein synthesis process can be broken down into 2 steps. The first step, transcription, takes place in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. First, an enzyme called RNA polymerase binds to a specific section of the DNA known as the promoter and starts to separate the 2 strands. One of the separated strands serves as a template for RNA synthesis. RNA polymerase then moves down the template strand, gradually assembling a complementary RNA strand known as the messenger RNA, or mRNA. The second step, translation, takes place in the cytoplasm. In this stage, the mRNA converts to a chain of amino acids that forms a protein. First, the mRNA formed in step 1 leaves the nucleus and binds to a ribosome, which reads the mRNA-1 codon, 3 nucleotides at a time. Each codon corresponds to a specific amino acid delivered to the site by tRNA molecules. The continuation of this process results in a growing chain of amino acids called a polypeptide. Finally, after translation, the chain of amino acids folds into a complex 3D structure called a protein.
Genetic mutations are alterations in DNA. They are mostly caused by incorrect amino acids being encoded in corresponding positions. Proteins have to be made into a specific shape, and even if the shape varies slightly, the proteins could not function properly. There are multiple types of mutations, which include nonsense mutations, point mutations, and silent mutations. Nonsense mutations, or stop mutations, halt translation, which causes the shape of the protein to vary, not forming properly. Point mutations occur in genomes where a single base pair is added, deleted, or changed. Silent mutations, a type of point mutation, do not affect the folding of proteins, thus is a neutral mutation. Genetic mutations can be beneficial, harmful, or neutral. Genetic mutations are the foundations of evolution. On the other hand, they can cause disorders such as sickle cell anemia, cystic fibrosis, and Huntington disease. Finally, we will cover DNA replication. Each time a cell divides, the information stored in the parent cell's DNA must be passed along into 2 new daughter cells. For both daughter cells to carry the exact same DNA as the parent cell, the DNA molecule needs to be copied prior to cell division. As the name implies, DNA replication is the process by which an entire DNA strand is replicated. Firstly, an enzyme called helicase cuts through the DNA strand, resulting in a replication fork. Then, a second enzyme called primase comes and adds a section of RNA known as a primer, marking the starting point for the construction of a new strand of DNA. After that, an enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end, and thus this strand is called the leading strand. The other strand, known as the lagging strand, which runs in the opposite direction, uses another method. The DNA polymerase can only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer, to which the DNA polymerase adds a short row of bases into the 5' to 3' direction. Next, the RNA primer is added further down the lagging strand, and this process repeats. Once the new DNA is made, the enzyme exonuclease removes all RNA primers from both strands of DNA, and another DNA polymerase enzyme fills in the gaps. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. The result of this process is two DNA strands identical to each other and the original.